Okay, everybody, this is our essentially last major group of organic compounds. We've done so far two. They have been carbohydrates and lipids. This is our third group of organic compounds, and that's proteins. There's one more group, nucleic acids, which we are only going to touch on a very tiny amount because we'll do so much of that when we talk about DNA later in the year. So this is really our last big group that we will tackle. This group arguably is the most important group of organic compounds, which is sort of a stupid thing to say because life as we know it wouldn't exist without any of them. But proteins, as far as what actually builds your body and makes the machinery work, and makes all the chemical reactions work as they should. Proteins are it. When you look in the mirror, what you see is protein and the results of proteins working. That's what you see. So proteins are extraordinarily important. Okay, and so to get something in your mind about what to think of when you think protein, just like when you think of carbs, you think bread. When you think of fats, you think butter and oil. When you think of proteins, you need to think meat. You need to think steak, and milk has protein, lots of protein in it. Fish, all of those things. Cheese. Okay, so now this is an extremely diverse group of compounds in terms of what they do, and better it probably is to say what don't they do. I mean, they are involved in almost everything in life. The probably most important thing that they do, that we will spend the most time talking about, is they act as enzymes. And if you'll recall from first year biology, enzymes are chemicals that speed up chemical reactions. They are biological catalysts, and we'll talk more about that later. But they are also structural. If you'll remember, when we talked about carbohydrates, they're mostly for energy, but they can serve structural functions too. These can serve structural functions. Your fingernails are made out of a protein called keratin, as is your hair. Collagen is really important in building structures in your body, like cartilage and tendons and things like that. They are very important as being transport molecules. Hemoglobin, you probably recognize that. That is the compound in your blood cells that carries oxygen, so it's a carrier, a transporter of oxygen. Aquaporin is another one. We'll talk about that a lot more later, but I bet I'll let you see if you can't figure out what aquaporin would transport. Cell communication, they're involved. There are hormones made out of steroids, which we talked about when we talked about lipids, but there are also hormones that are made out of proteins, so they're involved in communicating important messages from one part of the body to another. Insulin is an important protein hormone. So we'll talk more about the cell communication function. We're going to talk a lot about that later. Body defenses, they're involved in your immune system function. Antibodies are the most important things, and we're going to, again, talk all about this later. Your muscles are built on actin and myosin. When you eat steak, or any kind of meat for that matter, you're eating muscle. You're eating tons of actin and myosin. Okay, well, hopefully not literally tons, but there are also proteins that are involved in storage of energy and so on, and we can break down proteins and get energy out of them. We'll talk more about that later when we talk more about the energy process. Okay, this is where you really need to engage your brain right here. We know that proteins do a bunch of functions, and that's swell, but you need to know what they look like and how they're built. So this is back to the good old monomer plus a monomer plus a monomer plus a monomer equals a polymer. Okay, method of building a macromolecule. Remember, with lipids, this is exactly what we did with carbohydrates. Okay, whoops, sorry, should be a covalent bond there. If that's monomer, 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 or sugar, 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 this would be a carbohydrate. Okay, a polysaccharide. Carbohydrate. Yeah, my handwriting's really excellent there. If this is a protein, this is different monomers building up a protein, but it's the same basic plan to do it. Whereas when we did lipids just the other day, a lipid was really kind of funky and different. It didn't look like the little choo-choo train anymore. So this is getting back to this standard monomer gives us a polymer kind of idea. The monomers of proteins are amino acids. So just as when we talked about sugars, the monomers of sugars were the monomers of carbohydrates, of polysaccharides, were monosaccharides. The monomers here of proteins, okay, are amino acids. Amino acid, amino acid. I'm just going to AA amino acid. Okay? So we can see what we're talking about. Alright, so this is showing you exactly how two amino acids would come together, and this is exactly like everything you've done thus far. You take out an H and an OH, taking out water, and you end up with a nice covalent bond right there. And that is your peptide bond. This is just a covalent bond found in proteins. Remember, we had a glycosidic linkage in carbohydrates. We had an ester bond in lipids. Well, now we have a peptide bond right here in proteins. Same difference. It's all just covalent bonds linking two monomers together. So here's your two amino acids right here. We take out water doing a dehydration synthesis, and boom. Now, there are 20 different amino acids, okay, 20 different ones, which gives us the opportunity for a fair amount of variety when we are building these molecules. There's your dehydration synthesis. Remember, we take out the water. If we want to break down this polypeptide we're building, we just add the water back in. Okay, so with proteins, the term we give to a polymer of amino acids is a polypeptide, usually, because they are usually, if we've got amino acid, amino acid, amino acid, amino acid, amino acid, there's lots of peptide bonds connecting them, so we call it a polypeptide. It's just what it's called. Okay? A fully functional protein is usually not just one chain of amino acids, like right there. Usually a fully functional protein is composed of 2 or 3 or 4 of these polypeptides kind of clumped together. So a protein can actually be one or more of these polypeptide chains folded. And if you look here, these are different protein hormones, they're growth hormones, and this is a big old complex blob, and it's really made up of several different polypeptides jumbled up together. And it's not just jumbled, but you get my idea. Hemoglobin's the same way. Hemoglobin, this is one polypeptide chain. It's obviously been folded up, but that's one chain. This is another chain, this is another chain, and this is another chain. So that's 4 polypeptides, 4 of these amino acid, amino acid, amino acid, amino acid, amino acids, clumped together to make a complex molecule. So very large, very complex. Please note, proteins have complex 3-dimensional shapes, and those shapes are not just whatever, they are really important. The shape of these molecules is essential, and the molecules can do what they are intended to do. It's not just an accident they're shaped the way they are. They are doing a job. We'll talk about Rubisco later. So, shape is critical to function, exclamation point. Okay? All right, and we already talked about where the term polypeptide comes from. Okay, it's the term for that little bond, that covalent bond between two amino acids. Okay, so peptide means many peptide bonds. Okay, that's all polypeptide means. Okay, it may or may not be a functional protein if it's a polypeptide. Probably you need 2 or 3 of them. So this is our motif that I just kind of beat senseless, and I'm going to end up repeating myself right here. Just like with carbs, many monosaccharides, monosaccharide, give you a big old polysaccharide. Well, proteins are the same. Amino acid gives you a big old polypeptide. Okay? Many monomers gives you a polymer. Alright. Now, you need to know this. The structure of an amino acid. You should be able to recognize it. There's 20 different amino acids. You do not need to know the structure of each individual amino acid, but you need to be able to recognize generally an amino acid from some other compound, and it's not hard if you know what to look for. There is always a central carbon atom, and that central carbon atom is always bound to one hydrogen in that position. Now it can be drawn so that the hydrogen might be down low instead of up high, and you need to not let that fool you or throw you off. It just depends on how it's drawn. I think your textbook may draw them down low. I can't remember. So, in addition, so that's one hand. Remember, carbon's got 4 hands to hold. One hand's holding a hydrogen. The other hand is going to be holding what's called an amino group. If you'll remember, that's one of the functional groups we talked about earlier that gives a molecule, a hydrocarbon, some personality. The amino group always has a nitrogen in it. So that's one side of the carbon. The other side has a carboxyl group. Okay, that's COOH. COOH, if you just write it like that, but if you show where the bonds are, it looks like this. Okay, so we got a hydrogen, an amino group, a carboxyl group, which is an acid. Now, amino acid. Now you understand why it's called what it's called. It's got an amino group, an amino group and an acid group. Okay, a carboxylic acid. Alright, now what's the 4th hand holding? The 4th hand is holding something that is variable. Okay, and that thing that is variable is called an R group. I think that's R is for reactive, but it can also be called a side chain. This can be any one of 20 different molecular groups. Okay, 20 different groups. Any one of them can be placed there. So it varies. It's different for each of the 20 amino acids. That's where there are 20 amino acids. And this group, like any functional type group, gives each amino acid, this R group is going to give the amino acid its personality, its unique properties. Okay, certain amino acids characteristically behave certain ways because of what their R group is. All right, all amino acids have amino groups. All amino acids have carboxyl groups. All amino acids have hydrogen. All amino acids have carbon. So that's the same for all of them. The only thing that changes between them is this R group. Okay, that's what makes the difference. We need to think of these 20 different amino acids as being kind of like the 26 letters of our alphabet. Okay, we can put these letters of our alphabet together in different ways to make any number of different combinations of compounds. Of words, and like that. And then those words can go and make different sentences, different paragraphs, different books, whatever. These 20 different amino acids can go together in different combinations to make any number of different proteins, which can come together to make different characteristics in a living thing, different structures in a living thing, and different unique individuals. So it's very much like that. It depends upon the order in which we place these amino acids next to each other when we're making a compound. Okay? So the effect of different R groups, as you can see, some of them have kind of similar properties, so they get lumped together. These are all non-polar, okay, that we see here. And as such, they would tend to not be very attractive to water, so they're hydrophobic. And we don't need to remember all these funky names. We will work mostly with the abbreviations. We don't have to memorize those either. That's how we'll kind of refer to them. Okay. These are nonpolar and hydrophobic mostly because if you look here, there's a lot of covalent bonds that do not involve any oxygen. It's carbon-hydrogen, and we know carbon-hydrogen bonds are nonpolar. These tend to be polar. Okay. You can see that these have OH groups usually, or some kind. We talked about sulfur behaving like oxygen at the very one of the earlier PowerPoints. So these are tending to be polar and be therefore hydrophilic. These are electrically charged and tend to act like acids or act like bases. So those are some of the different characteristics that these R groups can give. Okay. We don't really need this. We don't really need this. We don't really need this. We do need this. Okay. I do want to mention to you about the SH group. We talked about the sulfhydryl functional group. When two sulfurs in a protein come near each other, like if neighboring R groups interact with that have sulfur in them, they can form really strong covalent bonds with each other called disulfide bridges between two sulfurs. So it looks kind of like that. It looks exactly like that, where you've got that covalent bond here, and it's really strong and acts to stabilize the structure of the polypeptide. So you should kind of remember that, and we don't need to talk about that right now. Okay. When you're putting together a amino acid chain to make a polypeptide, okay, it's like we've already talked about. We take out the OH of the carboxyl, now we know which end we're taking something from, and the H from the amino group. See, here's the amino group, here's the carboxyl group, and we butt them up just like two train cars, and we drop out that water molecule, and we have our dehydration reaction. Okay, so what you're getting, as it says right there, is a carbon-nitrogen bond. It's called a peptide bond. That's all you need to know. Okay, we've already talked about all this. Hopefully that's been covered. These proteins, polypeptides, do have one end that is quite distinct from another, because when you assemble the whole choo-choo train of amino acids, you do end up with one end that is an amino group and another end that is derived from that carboxyl group. And so they do have they have a sense of direction, a beginning and end, if you will. Okay? And the backbone, if you look just at the backbone, we're not going to consider the R groups for now. We're just going to look at the true backbone of this molecule. We see regularly repeating NCC, 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 and so on. Okay? That's the backbone of any polypeptide. Now, we already said that the shape is critical. Shape is really important in how proteins function. Their 3D structure is absolutely critical. And look at all these proteins here. They're clearly complexly shaped molecules. Okay? They can be twisted, folded, just any number of different things. So, how do we get there? How do we get to those complex folds? This is what you need to consider. The first thing that you consider in considering the shape of a protein is what order do its amino acids go in. That is critical. Just like the different letters of the alphabet is going to really change the meaning of a word depending on what order you put the letters. The order of amino acids in a protein or polypeptide is going to change how that polypeptide interacts with itself. If you put two polar, I'm going to put P for polar, or 3 polar amino acids right next to each other, they're going to be attracted to each other because they're all polar, and they might bunch up and make a little knot. Whereas if you had in the same position in an amino acid 3 non-polar amino acids next to each other, they aren't particularly attracted to each other. So your amino acid might be more, your protein might be more open at that point. So the order in which you put amino acids really makes a difference, just like the order of letters in a word makes a difference. Okay, so what determines, my kid is bothering me, go away. Alright, amino acid sequence that you get, the order in which you put those 20 amino acids, okay, is determined by your DNA, and that is how DNA builds you. It determines what your protein structure is going to be, and that determines what your structure is going to be. It's absolutely critical. Okay, so this, I really wish they wouldn't show it folded like this, because I'd rather, if we're just talking about the order of the amino acids, talk about it as literally the order of the amino acids. So imagine this is a straight line, and what you're seeing is we've got this lysine, valine, phenylalanine, glycine, arginine, cysteine. All these are different amino acids, and they're all in a very particular order. Okay, and if we change even just one of these, even just one, make a change or a deletion of it or whatever, it can have a tremendous effect on how this whole molecule folds and takes its shape. Okay? One amino acid can make a lot of difference. Let's talk about how much difference one amino acid can make. Okay? In sickle cell anemia, you've got val, his, lu, thur, pro, glu, glu. That's the normal the normal hemoglobin in a red blood cell. Normal hemoglobin polypeptide has that order of amino acids in it. Okay? And that results in really normal looking blood cells. However, valine, histine, lu, thur, pro, and we substitute at number 6 valine in place of glutamine. I believe that's glutamine. And it utterly changes the hemoglobin structure so that the blood cells themselves end up becoming misshapen, and that causes the whole cascade of symptoms we associate with sickle cell anemia, which is a devastating illness. Okay? So, why is it such a big deal? Well, this is a hydrophilic amino acid, this one is hydrophobic, and it reacts fundamentally differently than the correct amino acid does in that situation. Okay? Hemoglobin has 146 amino acids in it. One is wrong, and it causes this devastating illness. Okay? Now, that's the primary structure, the order of amino acids. What order do we put them in? And the DNA tells us what order to put them in. Okay? Now, secondary structure is just a sort of regular interaction between the regularly recurring parts of these amino acids. In an amino acid, a polypeptide chain, we always get this NCC, NCC, and these, the amino, the carboxyl, the amino, the carboxyl, that repetitive appearance of those functional groups in the long chain of amino acids. Okay? The carboxyl and the amino groups, as we're seeing here, 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 and there, okay, if you have them near each other, they will hydrogen bond with nearby partners. So, for example, right there, a hydrogen bond. If we had two sets of polypeptides near each other, hydrogen bond. Or if a single amino acid chain folded for some reason, like up here, if this went from being a straight line to being a folded line, and so we got some interaction of the amino acids over here and the amino acids over here, we could get this repetitive hydrogen bonding. There's a hydrogen bond that could result there. This is a hydrogen bond that could result here. And when we say hydrogen bond, we're just talking about this polarity. One thing likes electrons more than another thing, and it pulls them towards them and creates one atom that's polar, whereas another is not polar. And so we've got this slightly negative, slightly positive, and we get a polar interaction here, a hydrogen bond here, hydrogen bond here. It's weak, but it's stronger than nothing. Okay, now what you get, and this is again showing you the dotted lines are showing hydrogen bonds, what you get are regular repeating patterns of hydrogen bonding, and what that causes is different things depending on how it's occurring, but you can either get this thing called a beta pleated sheet, which looks very much like a child's fan that you might have made in elementary school and fanned yourself. You fold the paper, it's this accordion style folding, that kind of folding, or you get a spiraling helix, like a spiral staircase or a spring coil. Okay, so that's an alpha helix, and that's a beta pleated sheet. These are both very regular, predictable types of folding caused by hydrogen bonding between those regularly repeating amino carboxyl, amino carboxyl, amino carboxyl groups. Okay, now that's again showing you the folded beta pleated sheet, which would actually be, you'd have many of them, or the spiraling alpha helix and the hydrogen bonds. Now, tertiary structure, and is that we essentially take our child's fan or our coiled spring, and then we go and crunch it all up. Why would we do that? Well, the regularly repeating carboxyl amino groups are not the only things that interact. Those highly variable R groups interact as well. And when those different R groups interact, here's an amino acid, here's an amino acid, here's an R group, here's an R group. And if they're different, if they are non-polar, they're going to react differently with each other than if they are both polar. They might attract each other. If they're non-polar, they'll kind of be like, eh. If they are both negatively charged, they might actually repel each other. So they are going to react with each other very differently. And since you've got a bunch of R groups, when you have a long chain of amino acids, remember, every single one of those has an R group, and the different orders of them are going to really determine how these R groups kind of come together. And that causes what is called tertiary structure. It is an extraordinarily complex folding based on how the R groups are interacting with each other. So it's based on interactions between R groups, and there are several types of interactions. Alright, interactions between R groups. There are several types. Hydrophobic interactions are when two R groups or more are together, and the water that's in the solution in the cell around them is not particularly interested in buddying up with them, and so it kind of causes the chain to fold and isolate them and pocket them away in a fold of the amino acid chain. Now, hydrogen bonds and ionic bonds can form between R groups. Right here we see a hydrogen bond, much like those that we saw between the regular amino and carboxyl groups. We see a total ionic bond where two ions are being drawn together due to their opposite charges. And please look, all of this, each of these reactions is causing folding in this chain, and anchoring of those folds by these connections. Okay? Disulfur bridges, we talked about those. These are very, very strong because they're covalent bonds, and they will really hold the molecule in place very stably. Okay? So, quaternary structure, fourth level structure, is simply when we take multiple polypeptides that have their complex shapes and stuff them together. And that is what we see in this hemoglobin molecule we've already talked about. In this collagen molecule fiber, we see 3 alpha helices coming together to interact and so on. Okay? And at this point, we have a truly functional protein. Okay? We may not have a truly functional protein up until this point. Okay? Okay, we don't need to worry about that. So that's just a review of the primary structure determined by the DNA, the secondary structure determined by the amino and the carboxyl groups that are just repeating over and over, hydrogen bonds, tertiary structure determined by the interactions of the R groups together, and there's all kinds of interactions that can occur there, very complex, and quaternary structure, multiple polypeptides coming together. Now, we said shape was important for proteins, which it is. But if the shape gets messed up, what happens? Well, if a protein's shape gets messed up, we say the protein has become denatured. Okay? It's unfolded, it's misshapen, it's not what it's supposed to be. What could cause that to happen? Well, anything that can cause a molecule to alter its shape. Extreme temperatures that cause molecules to move fast can disrupt the bonds that hold them together. pH changes can, if you put in a lot of very reactive hydrogen ions, an acid type solution, you can get those hydrogen ions will interact with those various R groups and things in ways that are not predictable and disrupt the cohesiveness of the molecule. So temperature and pH, salinity, all of those are things, and we'll focus mostly on temperature and pH, that can alter the structure of a protein and thus alter its shape. And that destroys how it works, and we'll talk about that quite a bit in class, and we'll do a lab surrounding that as well. Okay? Sometimes proteins can return to their shape, sometimes they cannot. They are so damaged that they're done. Okay? These are just showing you different models of proteins. There's all kinds of ways scientists have of modeling them. Some use these ribbon structures. Some use these ball type structures like we saw with our little water molecules. They're just different ways of visualizing proteins. But at any rate, that's that. We're showing you a spider web here because why? Because that spider web is an amazingly strong protein. And that's it. And I'll see you soon. Thanks for watching.